Hello, my name is Philip Seitzer, and today I'll be introducing JContextExplorer, a program that is designed for cross-species genomic context comparisons. So, a little bit of background. All organisms, with the exception of viruses, contain a double-stranded helical polymer known as deoxyribonucleic acid. On this polymer, DNA, there are particular segments, which we call genes, that contain the instructions to create functional RNA chains, such as messenger RNA, which can then be translated into polypeptides or proteins. So when we talk about the collection of all of this DNA, all this genetic material, we're talking about the genome. In this example microbial species, the majority of the DNA is collected in this centralized bundle called the nucleoid, and there is also free-floating circular pieces of DNA called plasmids, which are also important for this organism to function. So, an analogy that is often used talking about genomes and genes, if we imagine the genes contain the instructions to make a single part, biological part, and a genome is therefore a parts list, the collection of instructions to create all of the meaningful biological parts. And here's an example, such a parts list. Okay, so what people have been interested in doing, especially in microbial species, is comparative genomics, where they sequence the genomes of various species, determine where the genes are and what they are, and then compare those genes across species. So in this case, we've got 3 organisms, organism A, B, and C, and as you can see, here's a segment of each of these organisms' genomes, bits of DNA, and you can see the placement of genes across all these organisms. A subdiscipline, a topic in comparative genomics, is looking at the genomic context, or the gene neighborhoods. In other words, how are the genetic environments of particular genes. And in this example, looking at gene 3, the green gene indicated by the arrow, we're interested in seeing how these surrounding genes, how the surrounding genetic environment changes across species, and therefore across evolutionary time. And as you can see in this example, looks like this gene has moved away from this group of genes to this group across these 3 species, across evolutionary time. So now, how does that all play into the JContextExplorer software package? JContextExplorer allows you to define different gene contexts, different genomic groupings, or gene neighborhoods. You know, here's one example where these genes are all grouped together into a common context, and then these are together, and then this set of genes contains, this group contains many genes all together. And there are various ways to do that, and depending on what you're interested in looking at, you may want to define these contexts in different ways. And as I'll demonstrate, JContextExplorer has many ways to do that. Once you have defined these genomic groupings, you may want to compare them. In this example, our gene of interest is this red gene, and the context is indicated by this red box. So you are interested in comparing each of these contexts to every other. So how does this context, A, compare to B, and how does it compare to C? How does B compare to A? How does B compare to C? And so on. When you have a set of things and you want to compare everything to everything else, you create a dissimilarity matrix, where in this case our objects are genomic groupings or genomic contexts, and we have to define some dissimilarity metric. How do we want to compare them? What's important? JContextExplorer has many features and many alternative ways to do that. But the way to read this object, you essentially see that A and B have a similarity level of 0.8. Often this is represented as a number between 0 and 1, where 0 implies completely, totally not similar at all, and then 1 is perfectly similar. We don't care about comparing something with itself, because that will always be 1, it's always the same. So once we have defined these pairwise dissimilarities amongst genomic contexts, we're often interested in taking all that data and organizing it in a meaningful way. So people use a technique called agglomerative hierarchical clustering to define a linkage tree, where the nodes of the tree are these gene groupings, and then these branch lengths describe the relative similarities of gene groupings to each other. Okay, so that's a brief background of what JContextExplorer is and how it works. Please see the next video, where I will take you to the website where you can actually download and launch JContextExplorer, and I'll highlight how you can do the things I've just explained.